could start now. Thank you.
Yes, thank you very much, Teresa. Warmly welcome to this webinar. My name is Anna Lennqvist, and the topic of today is something I personally find very interesting. And the issue of animal welfare versus chemical safety is always a very interesting and hot topic. But right now, especially in the light of the Green Deal and the chemical strategy, there is a worry that the increased demands for chemical safety will increase animal testing. And that is something we would like to elaborate around today. And I'm so glad that we have the speakers today, which are Kristina Rydén, who is professor in ecotoxicology and ecotoxicological risk assessment and regulation at Stockholm University. And we also have Mike Rosenberg, who's working for ECHA and has been there for a number of years and has recently been appointed and acting director of hazard assessment. And my colleague Teresa is here today to help us with the technical issues and also to co-host a Q&A session in the end. So you can at any time post a question in the Q&A function. The chat function is not available today, but in the Q&A, and we will answer them as good as we can in the end of this webinar. So first, I would like to give a very short introduction to ChemSec, and then I will leave the word to Kristina, and then Mike will talk, and then me, and after that is the Q&A session. And so please ask the questions you would like to have answered. So ChemSec, we are an environmental NGO. We are based in Sweden, and we are working for a toxic-free environment on the international level and mainly on the EU level. And as we are a small organization, we worked a lot together with other stakeholders, and we are involved in different EU policy processes in different committees and given our opinions on things. And we also challenge companies to improve their chemical management. And to do that, we also offer a few tools for making this transition. We also educate financial investors on how they can assess hazardous chemicals, both as an aspect of sustainability, but also as a financial risk. And by that short introduction of ChemSec, I hand over to Kristina.
Thank you, Anna. So, and thanks a lot for inviting me to this very interesting, I hope, and very important webinar discussing this very timely and interesting issue. So the question is whether we can enhance chemical safety and still ensure animal welfare. And to start this, I would like to just remind us that we are dealing with a super complex system. So we have maybe around 100,000 chemicals that are registered globally, and we need to that add metabolites, byproducts, transformation products. So a huge number of different chemical molecules that we're exposed to daily, and not just one chemical at a time, but also to complex mixtures of these chemicals. And these mixtures, they vary in time and in space. And the exposed, they are more than 100 million species, animals and plants and humans, of course. And these species can, again, be affected in very many different ways. So to completely grasp this risk system is almost impossible because it's very, very, very complicated and complex. But we do have a regulatory system that deals with these risks. And this is like a simplification of the system, an outline of some of its parts. So first of all, we have legislation that requires that we generate information about hazards and properties of chemical molecules. Different legislation. I will use REACH as one example, the legislation for industrial chemicals. So these legislations, they provide or make sure that we have hazard information related to a set of standardized tests that we have prioritized. And these tests, they provide information about certain toxicities, a selection of toxicities that we have prioritized in the system. This information then goes into the CLP rules, so the classification and labeling rules. They contain hazard criteria, and the purpose of that is to actually identify hazardous chemicals. And if a chemical then is identified as hazardous, it will be handled in a particular way. So we will provide hazard information in terms of like warning labelings and safety data sheets. And there will be downstream rules and restrictions on how we can use these chemicals, and all with the purpose of ensuring safe use in the end. So that's an outline of some important parts of the regulatory system for hazard assessment. It's not complete in any way, but it's an outline. And I would just also like briefly point out that also academic research can generate information about hazardous properties and of chemicals and feed into this system. And if we look at then REACH as one legislation that actually generates information about the properties of chemicals, we can see that it really promotes different ways of generating this information. So it says that whenever it's possible, information shall be generated by means other than vertebrate animal testing. So through the use of alternative methods, like in vitro methods, like different computer models for QSARs, but also we can use grouping and read across to utilize data that we have for one chemical to also assess other similar chemicals. Then if we move on to look at the REACH information requirements, and this is also a selection of or part of REACH information requirements, those that relate to human health hazards, we can see that REACH prioritizes testing based on yearly production volume or imported volume per manufacturer or importer. So we can see that REACH does not apply to chemicals below 1 ton, so no testing is required for those chemicals. And then test requirement increases as the tonnages go up. But we can see that the biggest test that is really mandatory for all chemicals is the subacute, or not all chemicals, but for the high volume chemicals is the 28-day study. So it's not like all chemicals registered under REACH will undergo a massive amount of testing. Also, if we look at these long-term tests, like the carcinogenicity test and reproductive toxicity testing, it's not mandatory even in the highest volumes. They can be required in the real high volumes, but it's not like every high volume chemicals will actually undergo those types of tests. If we turn to this part of the table, the different tests, we can also see that this is the selection of endpoints or toxicities that the regulation has chose to look at. But we can note, for instance, that there is no screening test requirements right now to identify endocrine disrupting chemicals. So endocrine disruption has been discussed in science for more than 30 years now, and it's really a health concern, but we still lack tests to actually identify these chemicals in a systematic way. So development is ongoing, but they're still not there. We also do not have any tests for immunotoxicity or neurotoxicity. These toxicities can be identified in some of these other tests, but it's not like we have a systematic approach to identify these hazards when we test. So this is a safety net that still has quite big holes in it. So this is just again to remind you about this picture, because we generate these data here in this example in REACH, with the hazard information should feed into the CLP criteria. So we'll also have a look at the CLP, the classification and labeling criteria. This is again a simplified summary of these criteria and what type of data that these criteria ask for, or that we need to sort of compare the data to the criteria. And as you see, as I have highlighted here, most hazard classification categories actually asks for animal data. Human data can also be used, of course, but if we have human data for a chemical, then the system has really failed. We do not want to have humans exposed to the extent that we find hazards and risks associated to that. But nevertheless, animal data is actually the preferred type of information when we use the CDLP classification criteria. We can also note that there are exemptions to this. So in vitro data is also allowed and can be used, or even physical data in some cases. But it's quite a lot focus on the animal data. So we may ask, why do we have such a focus on animal models? Well, we know that animals and humans are similar. There is a genetic similarity. And for the protein coding part of our genome, the similarity between a mouse and a human spans from 60 to 99%. So we are relatively similar in a genetic sense. We have the same organs in our bodies. These organs perform the same tasks in a similar way. So the body actually functions very, very similar. But we know, of course, also that we are not mice. So there are differences. There can be quantitative differences, like enzymes in the mouse that is more active than in the human, or vice versa. It can be qualitative differences, like receptors that we have, so they are conserved between species, but they have different tasks in different species. So sometimes in toxicity testing, the animal would be the more sensitive species, and sometimes the human would be the more sensitive species. But usually we do not exactly know what are the relevant differences for different types of exposures. But we have extensive experience with using animals as test species, and animal research lies behind major developments for human health. So many of the medicines that we take for granted today, like cancer treatments, HIV medicines, treatments for diabetes, Parkinson's, asthma, migraine, high blood pressure, depression, etc. They all originate partly from animal experiments. And also vaccines, different types of vaccines rely on that. Often we also use cell cultures to generate information. And cell cultures are really good to generate some types of information, like mechanistic information, for instance. Will this molecule bind to a receptor and things like that? And often in reality, we use a combination of animal models and cell systems if we want to really study an exposure and different effects. But cell-based models are not able to capture the complexity of the whole animal, the combination of these different organs and how they interact together and can cause effects in various parts of the body. So a cell model is just a tiny bit of what constitutes a whole animal with all its complex interactions between cells and organs. So it is important to understand that we cannot replace animal tests right now without losing a lot of important information. We lose all these interactions and we get only much smaller picture of what is really going on if we move from a whole animal to a cell model. And why is that so problematic? Well, in this system, the lack of data is treated as there is no hazard. So if we don't have information, say, from a carcinogenicity test, we have a chemical that has not been tested for carcinogenicity, the system assumes that this is not a carcinogen, and then it treats the chemical as there is no risk in terms of carcinogenicity. And we know, of course, that that is not true, because if we were to test all the untested chemicals for carcinogenicity, we know for sure that we would find a few new carcinogens. So this fallacy, no data equals no hazard, results in a systematic underestimation of risk. And it also means that if we reduce the information that we have access to, we also reduce the protection level. So less data will also make the system less protected. So this means if we return to this schematic picture of the system, if we have chemicals that are not tested, or certain types of toxicities like endocrine disruption, for instance, that we lack information about, this will be treated as those, there is no hazard in relation to those. We would say that the untested chemicals are safe to use. And this will lead to an unrestricted exposure of humans and the environment, and also uncontrolled exposure to these chemicals. And there we have a problem, because as I said, we know that if we were to test all chemicals for all endpoints, we would find more hazardous chemicals. And these are now used as if they were completely safe. So the key questions that we need to work with is how much and what type of data is needed to say that the substance is hazardous. And currently, as I said, just explained, less information means less protection. And that is not what we are striving for. So the chemical strategy, for instance, in some cases want to improve or increase protection, but in many instances, we at least want to keep the level of protection that we have currently. A more maybe controversial way would be to change the classification criteria and say that we can identify hazardous chemicals based on less information in a precautionary way. This would, of course, mean that we need to change the way which we generate information, but also the classification criteria. So it would really mean that we would have to rework or overhaul the current system. And as I said, to do this is also very controversial. There are stakeholders that really would not like us to consider chemicals hazardous based on less data than what is the case today, but it would be a possible way to do it. So how can we move forward and improve and be better at this? Well, first of all, I would like to conclude that animal models can currently not be replaced without significantly lower the level of protection. And that is not what we like. And I'm a scientist, but I have never worked with animal experiments, but I have worked with colleagues that do. So I know for sure that animal experiments are never done easily. They are always done for good reason. They are very expensive. They take a long time and take a lot of resources. They are high risk endeavors because so many things can go wrong with animals. They can catch disease or whatever. So they are never done without a really good reason. The use of animals is also, of course, highly regulated. But one thing that we should ensure is that we should use all available data. So the system right now has a preference for standardized tests performed by industry for regulatory purposes. But I also showed you that little square that said we also have academic research. So we should work, both scientists and regulators should work to increase the use of academic research in regulatory risk assessment. And that has some challenges. I will not go into detail about that today, but I think it's a very wanted development anyway. And of course, there is a lot of work and research ongoing to develop alternative methods. So we have 3D in vitro models, so cell cultures with different types of cells that mimic organs. We have computer chips that mimics also organs. We have these adverse outcome pathways that are being developed. So that is very promising. It takes a long time. And as I said, currently, none of these are actually able to sufficiently well replace animals, but they can become good supplements. And we, of course, hope that they develop further in the future. If you look at the legislations that actually generate regulatory data and the CLP, the classification and labeling criteria, we see that REACH really promotes the use of alternative methods to generate data, but that the CLP might not do that to a similar extent. It allows it, but in practice, it really asks for animal data. So one thing that I think the society and maybe the Commission should consider is to increase the focus on research and development of innovative test systems, because it's not just replacing one test with another. It's also how we combine tests into test systems and tiered approaches where we start with simple tests and make decision criteria that fit together and what can trigger more advanced testing and ultimately animal tests. But the important thing here is that the method development in terms of test models must also be developed in parallel with the legislation and the hazard criteria. So we need to find some kind of coordination between the test systems and the hazard criteria. So it's quite complicated in practice, but could of course definitely be done. Then the problem of the system saying that no data equals no hazard and that less information will lower the level of protection. Well, we already touched upon this. We can use read across and grouping to assess chemicals and hazards. And that is currently being done. And it's, I see and I think it is increasing, but we should promote that even more. That's a very effective way of using existing data. The more visionary, perhaps we also already mentioned, is to allow precautionary hazard assessment based on less data. That would require a change in the classification criteria and also a change in mindset in terms of how to use the precautionary principle in this system. Then, of course, we always hope for some young, super smart people that invent new approaches and methods to deal with the hazards and risks in this system. I mean, artificial intelligence is proposed in almost any part of society to improve things. And I'm not the person to say how that could be done, but I think it's an interesting intellectual exercise to actually think visionary about how we can do things or manage chemical risks in the future in a better way. So with that, I thank you so much for your attention. Thank you.
Thank you very much, Christina. I think that was very clear. And I would now like to leave the word to Mike from ECHA.
Good afternoon. I'm struggling with my computer. Give me 1 second.
That looks good.
That looks good. I assume you refer to the slides, but everything works. Fantastic. Good. Whatever time zone you're in, good morning, good afternoon, good evening to all of you. Delighted to talk about enhancing chemical safety and still keeping very much an eye or ensuring animal welfare. Let's see how I move the slides. Does it work? Like, yeah, it works like that. Sorry. Good. In the invite, there were a number of questions posed, and I am going at the end of the presentation give you the answer or our answer, the ECHA answer to these questions. But I don't try in the presentation to really substantiate each and every single answer because I only have, what, 15 minutes or so or less, I think. And it would take quite some time. But I will still promise to answer those. But what I did in the slide deck is to really give a view on what the role of the animal study is, can we do without, where not, and where the developments might lead to. So little bit, as Christina was saying as well, is that this is a wider context. This is not only about us, it's a very complex world. And I think one important institute or institution to mention in all of this is the OECD. The OECD plays a role in standardizing and standardizing animal tests by developing guidelines. Also doing a lot of work in actually standardizing approaches that are non-animal based. They ensure good laboratory practices so that once an animal study is conducted, that actually you can rely on it in the sense of the quality of it. And the other element that I want to pick up from this slide is the mutual acceptance of data. So once an animal study is done in a certain, according to the guidelines, according to the GLP, the good laboratory practices, then the different jurisdictions would accept a test, and they don't want, generally speaking, they don't want the test to be redone. So the whole structuring and organization of the animal studies is actually, and non-animal studies, it's the work where the OECD plays a central role, and it is very valuable throughout the world in terms of standardization, which makes them more reliable and predictable, but also in terms of acceptance throughout the world, and avoid redoing them whenever possible. When we move into the European context, there is a regulation, a number of regulations, and what I have on the slide is an explanation or a depiction of how REACH and the CLP regulation are supposed to work. It's a rich slide. It's also for you for later references to have a look at. But what you see on the left is what is it that these regulations actually try to achieve. And we're talking about a higher protection of human health and the environment, a better functioning of the internal market, increased competitiveness and innovation, and all of this ultimately supported with the promotion of alternative methods to animal testing. Now, at the right side of the slide, there is a whole bunch of activities, and I will not go into them in detail now, but it is a number of technical steps that are embedded in the regulation where either authorities have a role or industry has a role or both, and they all lead indeed to these four ultimate impacts. Then in the middle, there is a number of outcomes, which is a translation of an activity ultimately to the impact. And the degree of the compliance, so if people comply into our authorities and industry as well and other players, then we can get a high level of the protection, the better functioning, and the increased competitiveness, and the promotion of alternative methods and the use of alternative methods. So in this model, we've been working with REACH now for several years. And what REACH has brought is, one aspect that REACH has brought is clarity on which industrial chemicals are on the European market. And we've done that through different registration deadlines. And now we have a database, and we can analyze the database, and we can look what's going on with these chemicals. Now, what you see on this slide, again, it's a very rich presentation, but I will talk you through it, is the number of chemicals that have a severe health classification. So the C, the M, and the R stands for carcinogenicity, mutagenicity, or reprotoxic issues, and S dot are organ issues with organs after repeated dosing. So, and what is interesting to see here is that, roughly speaking, without the number of substances substantially going up, at least for the two top and the left bottom slides, is that you see an increase of chemicals that are actually known now to have these very severe effects. And you can imagine that having a chemical that causes cancer or reprotoxic toxicity, that's a serious issue, so to speak. And in Europe, at least, as soon as a substance is classified like that, immediately there are restrictions on how it can be used and not. So what the database makes transparent is that while we're working as authorities on the chemicals, there is an increase of chemicals that we know need further attention. This is fundamentally what this slide shows you. Another work that the authorities do is that we ourselves look at chemicals, and then we do a harmonized classification. Christina mentioned earlier the CDLP, to really establish at a European level what the appropriate classification is. And this slide here, it shows a table, and fundamentally it illustrates that in the last years, again, we've worked hard on making sure that we know the bad chemicals that deserve classification and therefore either need to be restricted or at least, if they are used, being used in a very safe and a very controlled manner. So to cut a long story short, there is a REACH mechanism that brings a lot of data to the table. Authorities are working on it, industry is working on it, and if you see the data, then we find more and more chemicals that deserve the attention. And again, with the note that we're looking at roughly the same pool of chemicals. It's not that the increase in the classifications that you see are new chemicals that we never saw before. So more information, getting more information on the table actually leads to a better insight on where are problematic cases, which leads to better handling the substances, which leads to a safer human health and environment situation. Now, I don't want to go too much on it because I think Christina already did a brilliant job in that, in the history of the animal testing. But when all of this started in terms of chemicals regulations, the animal models were the de facto to go to. And they were used to avoid testing on humans. They were regarded as better than testing on humans, obviously. And I'm not trying to make a joke. It is really like that. And later, another element is that a few tests are there to represent the entire environment, which is a development that came later. As I said earlier, from a regulatory point of view, from a system that tries to manage the chemicals and make sure that there is safe use of chemicals and protecting human health and the environment, there has been a lot of standardization. The GHS, the classification criteria, is one. I talked about the OECD, the mutual acceptance, and all of that system, that international system, is indeed based historically on animal studies. And this entire system, where we have all the opportunities and the possibilities now, it is there to protect humans and the environment against adverse effects. And you can say it provides a level of protection. Now, to quantify and qualify that is very challenging, but at least it sets a standard on how we protect human health and the environment. Now, obviously, we want to now, in modern days, we want to get out of animal testing as soon and as good as we can. And a lot of progress has been made. Now, I took this table, this picture, this figure from a report that we do every 3 years on the use and the promotion of alternative methods in REACH. And in simplistic terms, you see the darker gray is the animal test and the sky blue is the in vitro, so the non-animal test. And as you can see, through the years, there has been a switch in how from the animal to the non-animal way. Now, there's all kind of interesting details behind this and deadlines and all that. But I think one of the take-home messages is that, by integrating these new approaches into the regulations, there is an uptake, and industry is very willing to do them and to approach the non-animal alternative as not the alternative, but as the first choice to go to. And Christina already explained a lot of it. And I actually took this from the ChemSec website because I thought it is nice and elegant. I can quote it. And because if the European Chemicals Agency has to explain it, it becomes not always easy to follow. But I thought this was a particularly good, easy to understand definition of where the challenge lies. As soon as we're looking into effects that are involving larger parts of the organisms or longer times of exposures, then it's very difficult to do it with something else than an in vivo study, with an animal study. And at the moment, there are no replacements as such yet, and there possibly will be no replacement as such. But I will come back to that later towards the presentation. But it is said that if we want to have this similar level of protection, then at the moment we will need, for certain endpoints, for certain toxicities or diseases, we will need to use animal studies if we want to have a clear outcome on whether there are issues or not. also in the context of the regulatory system, as I explained earlier. Now, we in the authorities, we have received all these registration dossiers, which you can see on the left-hand top. That's our starting point. So that is the starting point for all our work. Now, I will not go into an awful lot of detail, but what this workflow shows you is that fundamentally we looked at what we got from the registering industry, and we see we slowly but surely are clearing up, as we call, the chemical universe to figure out where are the chemicals that need attention and where are the ones that are either safe or where are the ones that are really not a priority. So that's the illustration here. That's the work that we're doing. The first thing that we do is actually we don't look chemical by chemical, but we try to group them as much as we can, very crudely, very roughly. But we just want to make sure that when we look at a certain chemical, that we look at all the ones that are very similar, for several reasons. One of them is to avoid asking information on all of them if that would be needed. Now, once we've grouped the chemicals, we have a look and we see, do we know what to do with it? So do we have enough information to conclude on it? If not, then we go to the right top corner and we talk about dossier evaluation, substance evaluation, and the two assessments, where fundamentally we look at gathering new information or more information that we need to come to a conclusion. Does this chemical now really, or these chemicals as a group, do they need further work on? Now, on the bottom part of the slide, we go into regulatory terms. And it's fundamentally about classifying chemicals so that there is a European-wide understanding of the hazards. And then there are other mechanisms to do risk management on substances, for instance, by banning certain uses of the chemicals, which is on the low side. Now, we ourselves at the moment, besides grouping chemicals to avoid doing more testing than needed, is we're looking at non-animal information in the steps that I highlighted here. So when we do the first grouping work and the rough assessment of what is needed and all that, there we ourselves are also looking at all type of information that we have available to see, can we conclude on these chemicals, or can we conclude on what we need to do with them based on the information at hand. Now, we're not ready yet to do this at a very high level in the other processes in the bottom of the slide. I do predict that we will do more of it in the future, but one reason is that these are processes where we really conclude on chemicals in the sense of should they be restricted or banned or something like that, and then the whole legally sound and scientifically sound level of evidence that need to be gathered follows in a lot of cases having animal studies. Not in all cases, but in a lot. So that's why I highlight the top as the area where we do use a lot of alternatives, where in the bottom, not so much yet. This is a list that gives you some examples of the work that ECHA is doing. I don't want to dwell on it, also with looking at the time. I think one of the elements here that is very important to note is that we've gathered an enormous amount of data. That data is available, and it is available in different tools. So if we talk about avoiding animal studies, that's one area to really focus on. Use the data, use the knowledge that is already there, I would say. We do it internally, obviously, as well. The only thing I want to highlight from this slide is the last bullet point, where I talk about that ECHA is part of an international collaboration, which is called APCRA. And there we're doing work to figure out, can we actually move beyond the issue that also Christina highlighted, and I highlighted earlier, that at the moment, for some of these endpoints, there are no alternatives. And the way these alternatives are being developed, they probably will not be a replacement. Like you stop doing the animal and you do animal tests, you do something instead, and you will have the same outcome, because that's the challenge with the system behind the way we have it today. So one of the particular work that we're doing in this setup, which is international, as I said, with the US, Canada, some of our colleagues from Europe are there as well. And we really try to figure out, is there a way where we can use not one of these new approaches, but perhaps a combination of these new approaches to see, could they ultimately bring similar level of information? So Christina was talking about having less. We're talking here about, could we do something similar, not by having less, but having different information? And one of the particular projects that is ongoing at the moment, and it's a very rich slide. I apologize for it, but this is really, this at least makes me very passionate about talking about alternative methods. What we're concretely trying to do here is one of the animal studies that is, let's call it a workhorse for lack of better word, is the 90-day repeated dose toxicity study. So it is a study that is a repeated dosing of animals, and it gives you a whole range of information that either leads to classification or it informs you that there might be issues that you want to have a look at. Now, what we're trying to do here is not to replace it as such, but see if we could mimic this with other approaches that, and some of them do use still animals, but at a much lower scale than the classical study. But can we come up with something that at least has less animals that would ultimately give you the same information? Now, it looks like that we can, that the first signs of the work is that we could conceptually do something like that. Now, what we ultimately need to then, of course, figure out is it equally protective? And this is a bit where Christina was hinting at as well earlier. So this is, I'm very enthusiastic. We in ECHA are very enthusiastic about this work. It's not that we do an awful lot of it, because our main focus and our main mandate is not to dive into this, but is in executing the regulation as it is laid down. But the little time we have, we believe it's really worthwhile to look at these new techniques, and bottom line is to integrate them in the system as we have it today. Now, as promised, and I will speed up, as promised, I would answer the questions. So we believe there are actually many possibilities of understanding hazardous properties of the chemicals without using animal models. I mentioned the existing data as an example. There are other examples. Then, of course, one of the hot potato questions is, why do these regulators ask for animal studies? I have an answer here. It starts with that companies have failed to meet the legal requirements often. And the legal requirement is nothing more than actually to prove safety in a solid and robust way in terms of legal robustness, but also obviously scientific robustness. So, or we have a concern about human health and the environment. And then why we end up asking animal studies is a matter of being effective, efficient, and sound. We do look for alternative options before that, but ultimately we decide on closing the information gap if there is a concern. Now, we believe there are many options of reducing and even avoiding. I gave an example. Animal testing, they are already available. As I highlighted, more needs to be done. And in the future, it's really about looking at the alternatives and not replacing the animal study, but look at how these alternatives actually feed into an overall regulatory system to ensure a similar level of protection as we have today, similar or even better. And with this, I'd like to thank you.
Thank you very much, Mike. So I will continue. Bit behind schedule, but the two of you already said a few things that I had planned to say. So, do you see my slides now? Because I don't myself.
No, not at the moment.
No. Here. You see it now?
No, unfortunately not.
Oh. Yeah, okay, so I'll do another try.
Now it looks like something's happening. Yes.
Okay, so I wanted to start off a little bit in perspective again, and Christina and Mike also did it But we are in a situation where we have thousands of chemicals in production and in the environment, and we also have them in our bodies. And we have a huge lack of data as to what properties these chemicals have. And for the chemicals we know something about, that's really the top of the iceberg. But for those chemicals, we know that a high proportion are hazardous to human health and the environment, and that they are classified as such, so they are known to be hazardous. And there are more and more studies linking this to health problems in humans, like infertilities and cancers, but also animals suffer from the same problems, or also different problems. And we have a loss of biodiversity, meaning species extinction, partly linked to the issue of chemical pollutions. And I also went back and looked a bit at the numbers. There is, as Christina said, good statistics and information about the animals used in testing. So in the EU, and this is from 2018, about 11 million animals were used. Half of them mice, about 30% fish and 10% rats. And of those, about 18% used for regulatory purposes, and a minor portion of that, about 1.5% for industrial chemicals, which is REACH, and which is what we have been talking a lot about today. And when we talk about animal testing, the 3 R's often come up. And I think those are really nice principles or guiding principles about how to think about animal testing. So the 3 R's: replace, reduce, and refine. So meaning that whenever you can, you should replace animal testing. And if you need to perform animal testing, you should try to reduce that as much as possible. And when you do the testing, those tests should be refined so that we can get as much information as possible, and the animals should suffer as little as possible. And from ChemSec's side, we see that there are possibilities already today with the current regulation to do a lot to decrease animal testing. And actually, some of those actions also go hand in hand to what we think is necessary to also increase the protection level. And so I think that much can be done in both of these aspects at the same time. And some of these have been mentioned before. We have grouping, read across, better use of academic data, precautionary principle. And then in the end, of course, we also think we should develop non-animal methods further, and there are huge promises there. So going in to grouping and read across, it's what Christina also explained, that structurally similar substances often also share properties. So there are these groups of chemicals that are very similar to each other. Some current examples are PFASs, bisphenol, phthalates. And for those, we also have a history that one of those chemicals in that group have been well known to have a lot of problematic properties, have been regulated, and then replaced with a similar substance within the same group, later on proving to have the same hazardous properties, and then that one being regulated, and so forth. And this has now been recognized as problematic, inefficient, and called regrettable substitution sometimes. And to use grouping is a way to both avoid this regrettable substitution of a hazardous chemical with a similar one, but to also use read across, which means to use data from one chemical to also assume properties of a very similar chemical, is also what we can do to reduce animal testing. So we don't have to perform the tests on these very similar chemicals, but we can assume that they have the same hazardous properties. Yes. And the next one, which was also mentioned, was to better use the academic studies that are out there, that are peer-reviewed scientific data published in scientific journals. And that has up till now not been so much used in regulation. And so by regulators, these standardized OECD, often industry-funded GLP studies are easier to understand and to sort of put into the system and into the reports and dossiers and so forth for regulation. But there is this wealth of academic studies out there that could be much better used. And there is a shift in mindset now on how to better do this. And it involves, of course, efforts from both parties. Some scientists need to adjust their reporting a bit so that the studies and the findings especially are more easily understood by regulators. But regulators also need to be a bit more flexible in what information they are ready to use. And I just wanted to mention especially biomonitoring data and epidemiological data are fields which could be much more used in regulation, in my opinion, or in our opinion. Then we also have the precautionary principle and what we often refer to as paralysis by analysis. And being a scientist by background, you know that we can always do more studies. There is always so many interesting things that we would like to find out, and almost all scientific studies sort of end up with what could be done in the future. And it's also often the case that in regulation, when a regulation of a chemical is being discussed and there is some uncertainty, it tends to be easier for regulators to ask for more data than to take a bit uncomfortable decision and to regulate a chemical. And we from ChemSec's side think that we could do much more, much earlier, and sort of say that this is enough data. And with the help of the precautionary principle, we have the possibility to regulate when all data points in the same direction, even if data is not completely conclusive. And that could also help in avoiding further testing. And as Christina said, there is also this interesting balance here of interests, where there seems to be from industry a hesitance to regulate chemicals based on not so many studies, while it's not so nice to do studies if it is about registration. And yeah, we need to be aware of who is saying what and why. And then, of course, we need to develop non-animal methods further and support that development. We have in vitro, which means to use cell lines, isolated cells, and even parts of organs nowadays can be studied in isolation and give very good results. We also have better and better in silico, means in computer models for estimating hazardous properties based on structure and based on available data that are becoming more and more sort of intelligent and can do more. And to our experience, some endpoints, for example, persistence, which is a very problematic property, can be very well estimated without in silico with computer models. And that is also partly true for bioaccumulation and mobility. And that could be more and better used. And in conclusion, I think also much of what we are discussing today is planetary crisis and like global warming, biodiversity loss, all of these things sort of require the same answer. And that is that we need to produce less stuff, less hazardous chemicals. The things we produce need to last longer. We need to be able to recycle them. And all of this will also actually in the end lead to a less need for animal testing if we sort of go through with this transition. So we have only a very few minutes for questions, but we will turn to that now. So thanks for your patience.
Yes, thank you everyone for very interesting presentations. We have a number of questions and we will not be able to answer all of them. That's really clear. So I'll try to just summarize a bit of what I see and also linking it back to conversations that we've had with different stakeholders on what people think are interesting. So I'll start with a more future-looking question, and not really sure who wants to answer this, but think about it. The global production of chemical is supposed to double in the near future, and animal testing are very long processes. Can we really test the thousands of chemicals whose toxicity is still unknown if we keep performing the safety testing on animals and keep up with the toxic-free environment ambition of the chemical strategy? It was a bit long, but I mean, we do foresee that the use of different chemicals will go up. So how do you see that in the future? Is it possible for you, Mike, to have an answer on that?
Yeah, I can try to give an answer to that. I think, but this is the reason why we need to progress with all. There is almost, this conflict about you are in favor of animal or against animal studies, and then we don't discuss, but we argue. I think we need to move forward with the new approaches, and we need to figure out a way to integrate these new approaches if they are faster. I mean, the animal is one element. The other element is costs, speed, the number of, and therefore the number of tests you can do, etc. There are still quite some technical challenges, validation. There are all kind of hurdles, but we need to get this concept right on how can we replace what we have now with something equally good, which we then need to figure out what that means. Because indeed, there is a discussion on polymers at the moment. As an example, there is the whole mixture discussion. There are so many needs to understand better where we are exposed to, what are the implications of that. So we need to move away from animals. That's absolutely sure, with something that is quicker, etc. Having said that, at the moment, I think one of my slides shows you that the work we're doing using the animals where we don't have an alternative gives you still problematic cases on the table. The reality is you cannot solve it overnight. This will be a long stretch with so many questions and so many things to answer. So it is about looking forward, is more about a debate on how do we integrate these new approaches rather than having a polarization of you are either in favor or against, because that doesn't bring an awful lot ultimately.
Yeah, thank you. Yes, Christina, do you have something to add there?
I can also comment. I totally agree with Mike, of course, about the future, but I also think we can reconsider. And one thing that, at least in theory, is a low-hanging fruit is the prioritization criteria, which is now production volume per year per manufacturer or importer. And that prioritization criterion is based on economics. So large production or sales would carry the cost of more testing. And that has nothing really to do with inherent hazards. So consider a system where we sort of start by sorting the chemicals into hazard categories based on like structure activity or something, and then adjust testing just according to other criteria more related to the molecule and the expected properties of the molecule, like a QSAR or something, instead of these just blunt volume. There is indeed information and practices that show that the high volume chemicals are less toxic or less hazardous than the low volume chemicals, because you don't become a high volume chemical if you are super hazardous. So this is a little bit counterproductive and at least in theory, a low-hanging fruit on how to improve a tiered approach for testing, which would be more optimized and provide more information per unit testing, so to speak.
Thank you. I realize that we're already at the time limit, but I wonder if it's okay that we just go on for about 10 minutes because we haven't had time for questions. Is that fine? Okay. Thank you for that. I'm just going to bring up a question that we see a bit in the chat, and we also heard from different places, and it's how the REACH regulation corresponds to, for instance, the cosmetics regulation. So I wonder, maybe Anna, if you could go into a bit, how can we make sure that these are aligned, and what is the actual issue that we see here?
Oh, that's a good question. I think maybe also Mike wants to come in there. But as I have understood it, animal testing have been out of the question for cosmetics and ingredients for a while. And there have been alternative methods instead, and I think quite successfully. But then there are also ingredients that are used both in cosmetics, but are also under REACH and for industrial purposes. And there in the REACH regulation, there have been requirements for some of the same chemicals that are used in cosmetics as under REACH for further testing. And that has been seen as problematic for the cosmetics industry because they don't want to use ingredients that have been tested on animals, even if they have been that in another context. And so there is a sort of apparent conflict there. And I don't know, Mike, if you have worked anything on that or want to say something more there.
Yeah, I'm going to repeat in different words what you said. I think the issue is that, so the cosmetics regulation talks about the end use of cosmetics. So when it goes to the actual consumer using a cosmetic, then there is a requirement of or the animal test ban. At the same time, many of these chemicals are also part of REACH, and in some extreme cases, they are being manufactured and then being used as cosmetics. And as you said, many cases, actually, these chemicals have all kinds of uses. So, and I think but it's another example of a debate where the nuances are often too complicated to make good news stories. So that's a bit of a pity. The nuance is the following: that if a chemical is used under REACH, and REACH tries to protect, also, for instance, workers and the environment, then that particular chemical, if you look at, there might be workers' exposure, and there are some examples that led to further discussions in court, etc. But there are then considerations to make sure that these workers who are exposed to completely different concentrations in a completely different way, not necessarily by their own choice as a consumer, so they fall under the legal construct of REACH, and then there might be a need to ask for studies. And sometimes these are animal studies, not always. And the focus, that all has to do with the area that REACH tries to protect and where the mandate stops and where the mandate ends. And then, indeed, there are the situations where there is an overlap, which leads to confusion and a lot of discussion. The cases where we've looked at the chemicals, there was always an element of exposure to, of potential exposure to, for instance, workers, or cases where a chemical is used for 30, 40 other uses, not only cosmetics. So, and that is where there is, let's call it the overlap. In terms of implementing what the legislator said, we're confident in the cases that we look at. We are also confident that there is actually a need to look into it, but we do recognize that it leads to confusion on what does it mean now for cosmetics, obviously.
Yeah, thank you. I mean, it's clear that this needs to be worked on, and it shouldn't be penalized to have more information that can actually provide better protection for human health and the environment. Thank you. We have a lot of questions. I'll pose a last one. I'll try to put a few together. There is the impression from some stakeholders that there is this higher bar or increased demand for animal testing now, and that there's been a shift here. And there are questions about what is the position of registrants regarding the acceptance of regulatory action based on alternative methods, such as grouping or other measures that you have already mentioned. Mike, maybe you can give some answers to that reaction from stakeholders.
You should ask industry, you shouldn't ask me. I think it's for. I mean, this is the challenge ultimately, because I think as soon as we start to use we don't easily, from an authority side, will use alternatives for these very complex studies, because most of them are actually not suitable. Sometimes they are suitable in a very particular context. I think industry then has a tendency to argue on a scientific level. So it's not against the principle. Industry doesn't say you cannot use the num because of the num. They will argue, from their scientific perspective. I think that's safe to say. But this has been really very specific topical cases. Now, I think if the question would be, let's open all of this, let's not have any validation, you can use whatever you find in literature, you can use it to provoke a bit. There are other questions related to that as well. I don't know how industry would react. I think, as I said earlier, at the moment, there is a high level of standardization. There is a high level of validation. It's all implemented in regulation that gives everybody legal certainty. So we know that if we have one of these guideline studies, that often these, hardly ever the study is questioned. We also know that industry might go into some of the details of that, but questioning the entire concept of the study doesn't exist anymore. So I would wonder, I think it makes the whole regulatory process, perhaps, people sometimes complain that it takes so long to before a chemical is restricted. But I think thanks to the standardization, at least that part is not discussed so long. So, but I'm giving a very, very biased, because we're in ECHA, we don't make the regulation, we execute the regulation. And it's not that I want to get rid of the answer, but our focus is very much on the process and legal certainty, etc. So that's why you get the answer that you're getting, in sense of it's really it's really a great good that we have this high level of standardization and predictability, not in a toxicological sense, but more in what's going to happen if you have the study, if you didn't have the study. That's also when we look at the dossier and we think there are problems and it's not compliant, we ask as a first principle the study. Now we say you can always come with different, but it's for the same argumentation of clarity. Then everybody is clear what needs to be done to get this situation right, and I think that's incredibly valuable in the whole chain.
Yeah, thank you, Mike. I realize it's a very difficult question to answer. I will give the word back to Christina first for some final remarks and additions, and then back to Anna to conclude.
Yeah, I also comment on this last question, because I think whenever we touched upon this already, but whenever we want to replace an existing test or a suite of tests with something else, I think it's really important that we think in terms of the protection level. So tests can go wrong in different ways. It can be like too sensitive, so generate a lot of false positives. We identify hazardous chemicals that are in fact not hazardous, or they can generate false negatives. So they are insensitive, so they miss substances that are actually toxic. And ideally, we would like true positives. We would really identify the hazardous chemicals, and the benign ones should not be identified. So we need to sort of, for every change that we do in terms of test requirements and hazardous criteria, discuss in those terms. Do we increase protection or do we lower protection? Sometimes I see the discussion focusing on mere numbers. How many can we cope with? How many more will be regulated and so on? But I would like to extend that to also actually really address the protection level. And that is, of course, difficult, because what to compare to and how to do that, but it's still conceptually very, very important.
Thank you. Great last words there. And Anna.
Yes, thank you very much. And thanks for hanging on an extra 10 minutes. I realized we could have had easily half a day to discuss these issues. And huge interest also from in the Q&A session and many participants. I'm glad to see this discussion ongoing, because it really needs to happen. I think there are a lot of things that can be done to improve the situation. And it's also very important to have this discussion a bit more difficult word, a bit more nuanced than saying no to animal testing or yes to animal testing, but it's so much more than that, and there are so many things we need to consider. So thanks again very much, Christina and Mike, for being here, and thanks to all of you that have listened. And this discussion will continue on email and other for us, I'm sure. So have a nice evening, all of you.